Hi to everybody that is watching this video. My name is Matteo, and in the next 20 to 25 minutes, I'll guide you through and explain the paper I've chosen and the associated research, which is covering, in my opinion, a very fascinating but also particular topic, which is the one of detection of surface biosignatures for the search of life in exoplanets. The study spans over a period of more or less 3 years by a team of scientists that are located in a multitude of universities, mainly University of Lisbon and Cornell University in New York. I think we can start. So upcoming ground-based extremely large telescope, as well as the James Webb Space Telescope, which most of you, I assume, are familiar with, or at least have heard of, can search for signs of life on extrasolar planets. However, the detection of planets in the habitable zone does not guarantee habitability. Other than atmospheric assessment, surface biosignatures are a strong tool that can be used to identify and assess the presence of life on exoplanets. And future large space-based telescopes, so telescopes that are not grounded on Earth, such as the large ultraviolet optical infrared surveyor, which you can see here on the right part of the slide, and the Habitable Exoplanet Observatory, which you can see here, are being currently in design and formulated to explore these biosignatures or surface biosignatures in both in the atmosphere and on the surface of exoplanets. Pigments of microbes and bacteria are on Earth signs for life on the surface, and they have a unique reflectance that can be used as a signature to identify living organisms on exoplanets as well, and a color library based on Earth observations, so establishing a color catalog based on just the observation we can do on our planet can provide a guide for the search for surface features of extraterrestrial life on other planets. The data retrieved from the Earth unique reflectance signatures of microorganisms can therefore be used to search for life on other planets. And what examples do we have already here on our planets? So as you can see here from this image, this pink color given to this lake is attributable to the bacteria of Holobacterium genus that require an environment with a high concentration of salt. This is a purple species is very light sensitive and has a very light sensitive protein, which this protein provide the chemical energy for the cell by using sunlight to pump protons out of the cell. Another example, as you can see here, this is a space picture of the Coral Sea. We see these lines here. So these are Trichodesmus bloom. This is Filamentus cyanobacteria. They are especially found in nutrient-poor tropical and subtropical ocean waters. And this bacteria fixes atmospheric nitrogen into ammonium. And one last example, this is a satellite image of a large bloom of Nodularia swirling in the Baltic Sea. This is a bacteria belonging to the genus of the Filamentus nitrogen-fixing cyanobacteria, more commonly known as a green algae and these bacteria mainly occur in salinic waters. So you see that we have example on Earth of bacteria, of microorganisms that have very particular albedo and have a color reflectance that allows extraterrestrial observation and detection of life. And a color catalog exists, but is not complete. So there have been mainly, I would say, two studies or two research that are worth mentioning. So the first one is the one of Edge and Kaltenegger, sorry, published in 2013. So what they did is a little bit the opposite and coming up with a color catalog. So they actually used albedo of extremophils to explore the characterizations of the Earth surfaces in extrasolar planets. And what they did is that they detected rocky Earth analog exoplanets based on the surface signatures of these extremes forms of life, but they did not come up with a proper color catalog. This has been done 2 years later. Mr. Hedge was still in the lead of the research, and what they did with the team is that they came up with a database of the reflectivity spectra of a set of more or less 140 microorganisms from different environments. And this catalog cover the visible and near infrared wavelength portion of the electromagnetic spectrum. But again here, no organism from a frozen environment has been collected. So there is a lack of knowledge, and this color catalog lacks the color reflectance of bacteria and microorganisms that live on ice. But why ice, the ice ecosystem, or why this is so important? So a great abundance actually of pigmented microbes have been found in a highly diverse ice microbial communities. So living actually on ice, and various researchers actually isolated them from Arctic or Antarctic samples, suggesting that icy worlds orbiting other stars or icy moons may provide promising targets for the search of life. Life actually may also have started on Earth in ice. This is the so-called cold origin of life hypothesis. And a diverse set of microorganisms has been detected in the ice sheet of a lake in the Antarctic, as well in the permafrost, making both environments good analogs for the icy moons, like, for example, Europa, Enceladus, or Ganymede. These are moons that have an icy crust and the possibility to have also liquid oceans beneath the surface. So coming to the goals and the methodology of the research. So as we talked, and as we said, a database of biota in icy environments on Earth is definitely a critical tool that will enable the search for life on icy exoplanets. So the goal exactly of these researches is to expand the existing color catalog that I mentioned before by including ice biota reflection spectrum. So including the reflection spectrum of bacteria and microorganisms living on ice. So the methods. So the team of researchers collected 80 microorganisms from ice and water in a remote location in Canada, and they identified each of these by using RNA and the sample preparation in all the culture were grown at a room temperature depending on the isolate, the time required for the growth varied between 24 hours to 1 week. And all the procedures during the culturing process occurred under, of course, sterile conditions. And then the team took this bacteria and this biota and deposit them, the cultures on a 25 millimeter plain white mixed cellulose filter. And a spectrometer was used to measure the reflectance of the samples. And this is exactly the same device that has been used in the previous catalog, so to avoid any bias between the two studies. The same samples the same sample of this study were measured at 2 different times. So immediately after being deposited on the filter, so a fresh culture, and also after 1 week. So this allowed the sample to dry, so that there was a measurement under fresh conditions, or still with water, and one measurement after 1 week in dry condition. And this is an image that depicts so this filter, and with the bacteria on it, you can see that there are already a variety of colors ranging from see-through on the bottom right corner, to green, to more yellow, to reddish, and on orange. So we have a large variety of pigments that characterize this sample. So this is actually the main and most important result, the main result of the research. The figure here, this figure shows the light reflection per wavelength. So light reflection on this axis, wavelength on this axis. So it is possible to identify the various color signatures of the biota isolated, and these have been grouped in their colors and the colors reflected. So we see here more yellow. We see here pink, reddish pigments or reflectance. We see white. We see orange, and we see green. And there were also unpigmented, so more see-through organisms that acted as a biological control for uncolored organisms. So we see here that yellow, orange, red carotenoids are the most frequent reflection in the sample group. So when we zoom into the figure, so the left column here represent fresh samples. So the first that was taken right after the microorganisms were laid down on the filter. And this left column represent more the dry samples, so the one after 1 week. So we can see that the dry samples actually provide a stronger reflection. So you see here the graphs for the same wavelengths, the lines are at a way higher level compared to the wet sample. And also, if we zoom here in these 2 columns here, so if we look at the wavelength between 35 millimeters, 35 microns and 1 micron, 0.35 microns and 1 microns, we see that this is the range with the highest reflection. So this is this part of the graph reported. So we see that at this wavelength we have the highest reflection for, I would say, almost all of the sample and all of the different carotenoids that characterize the sample collected. So what can we learn from these findings? Why are these results important for us, and what do we need to understand from these results? So first of all, the carotenoids are a signature of life on ice. So we see that icy biota actually have pigments and carotenoids and they provide an albedo which can be observed. And these caretonides are not limited to a small taxonomic group, but as the research highlighted, they're present both in photosynthetic and non-photosynthetic groups. Carotenoids have, as you might know, play a key physiological role for organisms in extreme cold environment. They protect from radiation, photooxidation, lack of resources, and dryness. Second thing, very important, we saw that dry samples actually provide higher reflectance. So dry samples reflect stronger. This finding is backed up also by other research. There is a research of Bake and colleagues of 2020 showing that dry samples make the detection of pigment biosignatures way easier. So this indicates actually that water absorption or the presence of water actually reduce the reflectivity of pigments. Why? I mean, I don't want to go too much in detail, so in case, I'm really happy to answer this question in the comments. And also, I think that I mean, the dry microbial cells are therefore an interesting target for the search of life. And especially these are well present in dry and highly irradiated planets. So planets, for example, that do not have that actually have a high level of radiations. And such a color catalog developed on Earth, so with the sample of bacteria coming from Earth, actually lays the ground for future missions that will observe exoplanets surfaces, and that instruments that will observe the albedo of exoplanets. Actually, we have a couple of instruments and missions that are currently underdeveloped and planned to launch. So there is this mission. Sorry, I thought I had the name of the two, but not in the slides. So, so the missions such as the mapping imaging spectrometer for Europa, which is this one here on the right. This is a mission that will fly on NASA Europa Clipper, which is an orbiting satellite in the late 2024, to explore the potential signatures of extremophiles that are in the icy surface of Europa. And also here, the Europa, I mean, this is not really a clear image, but the Europa Lander spectral imaging experiment. So I've not been able to retrieve a proper image of this instrument because it's still under development. But this is also another project that is currently under development and ongoing to measure and observe surface biosignatures on Europa. And coming back to the MISE, so this instrument here, this project here, this is actually an imaging near infrared spectrometer that will, as I said, be placed on board of the Europa Clipper mission. And this will observe the Europa, which is Jupiter's moon, and will observe its surface composition and relate it to the habitability of its internal ocean water. So I think to conclude, a critical perspective and a little bit my take on the research, and also what do I think the future outlook will look like and where future research will need to focus on. So the Earth atmosphere and magnetic field definitely shelters it from solar radiation. But these actually do not hold true for many exoplanets, as we know, and that most of them do not have an atmosphere. So, for example, Europa receives 1,800 times the average annual dose of radiation that are experienced by a human on Earth. So therefore, the reflection spectra must definitely need to be adapted or reconsidered when exploring this type of planets that are subject to an intense level of radiation, because the albedo will definitely be different from the one on Earth. Because yeah, I mean the radiation destroy organic compounds way faster, and this will definitely impact the albedo of these planets that do not have an atmosphere or are not protected from high level of radiations. Second, then also the measurements were carried in, as I said, Earth conditions, and also focusing on Earth-based life. So my question is this catalog really applicable to all exoplanets? I mean, yes, I mean, Earth, water-based life is definitely an option for extraterrestrial life, but it's not the only one. We know that silicon is a good molecule from which life may arise. So how would a silicon-based life alter the result? How would silicon or methane actually influence the albedo and the reflectance of these pigments? Therefore, as next steps, in my opinion, necessary to achieve a high accuracy and to have this catalog, color catalog, really applicable to the search of life on exoplanets, I think it would be crucial to get rid of this Earth bias and to determine how this confounding effect of our atmosphere influence the reflection spectra. So yeah, I think here I arrived to the end. I thank you very much for watching, and I hope you understood everything. In case of any questions, of course, I'm happy to answer them in the comments below. Thank you.